Okay, welcome to chapter 17, the nervous system. The nervous system is made up of the brain, the spinal cord, and all of the nerves in the human body. And its job is to coordinate and regulate the function of every other system in the body. So the cardiovascular system, the respiratory system, everything in the body is controlled by the boss, which is the nervous system. The boss. All right, here in this picture, we've outlined the brain, the spinal cord, and the nerves. Okay, the nervous system has two parts: the central nervous system, or the CNS for short, and the peripheral nervous system, or the PNS for short. All right, the central nervous system is made up of the brain and the spinal cord. And here the central nervous system is isolated in this image. You can just see the brain and spinal cord representing the central nervous system. The central nervous system's job is to receive, process, and transfer information. The peripheral nervous system is composed of all of the nerves that are outside of the CNS, meaning all of the nerves that are outside of the brain and spinal cord belong to the PNS. Okay, the PNS is divided into two subdivisions: the sensory division and the motor division. Okay, the sensory division carries information towards the CNS. And a good way to think of it is like this. The sensory division, its job is to sense information, right, and carry it to the central nervous system, which is the kind of the center or the primary component of the nervous system. Okay, the motor division, it takes information away from the central nervous system. All right, so in other words, okay, sensory is going to be carrying information towards the CNS, the brain and spinal cord, okay, depending on the signal. And the motor division is going to be carrying information away from the brain and spinal cord and towards the body to elicit or carry out some sort of response. Now, let's talk about the cells of the nervous system. Okay, there's two primary cells that make up the nervous system: an actual neuron and the neuroglia. So let's start with the neuron. All right, neurons are cells that can transmit a nerve impulse. A nerve impulse is technically going to be an electrical impulse. So the reason why our nervous system can transmit information so fast is because literally within these neurons there's electricity that shoots through our nerves, basically. I mean, it's more controlled than that, and we're going to learn the details of it. But this is completely specialized to neurons. No other cell in the body can transmit electricity, or aka a nerve impulse. All right, so neurons can transmit nerve impulses between the different parts of the nervous system, to and from parts of the body, and basically the entire body. Every part of the body has some sort of neuronal connection that goes back and forth from the brain to that part of the body. For example, our urinary bladder has neurons that connect to and from the brain. Okay, our heart has neurons that connect to and from the heart to make sure that it's pumping. All right, so neurons make that happen, and they're incredibly important. And then We have secondary cells, which are called neuroglia, and these guys are just basically there to, they have a big role. They support neurons, they nourish neurons, they help maintain some homeostasis, and they make something called myelin. Okay, myelin is a structure within within a neuron that help to aid the signal transduction. So here we're going to look a little bit closely at this image. Okay, this is a neuron, and it's kind of, this is the cell body. This is the beginning of the neuron, right? Okay, these little dendrites are sticking out, and we'll learn the full anatomy. And then all of this that's going this way, okay, is part of the neuron as well. And then you'll see to the around the neuron, there's these extra little cells like the microglia, the oligodendrite. Okay, those are these little cells, the astrocyte. These are all neuroglia cells. If we were to look at neuronal tissue under the microscope, this is what you would see. You would see a neuron, and you'd see the primary body. There's the nucleus, and you'd see some of the dendrites, and then these little tiny dots like here, everywhere basically. These specks. Those are the neuroglia, and basically they're small, but they have a vital role. And this is actually a great picture because it's showing you that's a neuron, that's another one, that's another one, this is one over here, and then all of this other stuff, all these little tiny specks, those are the neuroglia cells. Okay, so let's look at the neuronal anatomy. A neuron has three primary parts: the cell body, okay, dendrites, and then the axon. And it actually has a little bit more that I'm going to discuss later on, but let's start with that. So the cell body is the main part of the cell, which houses the nucleus. So it's right, this kind of mass right there. There's a cell body. Okay, and then these little small branches that connect to the cell body, the shorter ones, all of these. Okay, these are called dendrites. Okay, and now this whole long region over here, this is called the axon. And then eventually the nerve is going to end, and it does have this very specific end called the synaptic terminals. Okay, now this is incredibly important, and if you're taking notes on what you should be doing, you want to know that the pattern of neuronal transmission is as follows. Now, information comes in through dendrites, okay, so it's coming this way, and of course it's going to be coming from another neuron, and it'll go through the cell body, okay, and then it will move down an axon away from the cell body, and then eventually the synaptic terminal will transfer the signal to the next neuron. Okay, so electrical signals move this way. All right, down an axon. Okay, now neurons, there's different types, and they're completely specialized for communication. So we have sensory neurons. Okay, these guys are going to be found in the PNS, where they receive information from your peripheral nervous system, and their job is to transmit it to the CNS. Okay. Now, then we have interneurons. Interneurons stay inside of the CNS, and their job is to transmit information between different components of the CNS, the central nervous system. And motor neurons, Motor neurons are found in the peripheral nervous system, and they take information away from the CNS. Okay, now let's look at the second image over here. Okay, now these are sensory neurons, and so this is just one example. Okay, so let's say that this is going to come, these receptor endings, the sensory neuron over here, these receptors are going to detect information. Let's say it's information of you put your hand over something hot. Now you have sensory receptors in your hands, in your skin really, that's going to pick up that information, and it's going to carry it towards the central nervous system. Okay, within the central nervous system, so at this point over here, let's be very clear. Okay, this side is the CNS. This side is going to be the PNS. So it's going to, the sensory neurons carry information towards the CNS. Now these are interneurons right here, and interneurons will pick up that information. These are interneurons that are inside of the central nervous system, and they will elicit a response. Typically with heat, it's going to be a reflex. Okay, but they stay inside of the CNS. So think enter within the CNS. Okay, but they connect directly to motor neurons. Now, motor neurons will carry that signal away from the CNS towards the PNS and then elicit a response. In this case, it would be a reflex to move your hand away from this hot fire. All right, now this happens in like nanoseconds because, again, these neurons are carrying information through electrical impulses. All right, now there's three basic types of neurons. The first one we have is called a bipolar neuron. And these are typically our interneurons that are going to be in the CNS, and it's called bipolar because of this. Okay, so here's the central body in the middle. Okay, and then it has two poles, one going this way, one going that way. So it has these two axons, and then the dendrites are over here. Okay, now a unipolar neuron is going to be a sensory neuron, and so information is going to come, from one side, get processed here, and then sent from this side. Okay, so if you look at this picture, the first one, notice that the cell body kind of is sticking out, all right, because there are different types of neurons, and that's really that. So it's called unipolar because notice the cell body, if we trace it around, it's going to have one pole. Okay, and then of course it'll have a signal from one side, it'll get processed here, and then out this side. A multipolar neuron, okay, that was unipolar. Okay, these are going to be our sensory. Multipolar neurons are motor neurons, and they're called multipolar because of the structure. Notice how this is kind of the more typical one that we're used to kind of seeing pictures of, but there are different types. So it's got these multiple poles, right? And those are technically all dendrites. And then, of course, the axon and synaptic terminal on that side. All right, so we have bipolar neurons, which are interneurons that belong in the central nervous system. Unipolar, aka sensory neurons, which carry information from the PNS to the CNS, and multipolar neurons, which carry information from the CNS to the PNS. Now this image is showing us an example of what we just saw actually, but it's showing us in in a little more detail. So what we're looking at here, this is a coronal section, this is a spinal cord, and actually this is what the spinal cord looks like if we were to slice it open. This is gray matter, and it kind of looks like a butterfly shape. This would technically be white matter. Okay, now let's see. Okay, what's going on in this picture is this is a muscle cell. And the idea here is to show how the three neurons work, how they work to initiate a response and carry out a response. So let's go ahead and follow through. So this is a muscle. Okay, and you see information coming from every single muscle has tons of neurons. In fact, we're going to learn quite a bit about, when we get to our muscle chapter, about how muscles contract and uncontract through the control of the nervous system. Okay, so now this muscle is giving off a response, and that response will be carried by one type of neuron only, a sensory neuron, because their job is to carry information from the PNS, which is everything outside of the brain and spinal cord, towards the CNS. Okay, and it will synapse or connect to right there to this neuron inside of the spinal cord. So not every neuron needs to go to the brain. In fact, this one just will, the spinal cord is capable of responding because it's structured that way. And then you see this interneuron right here. It goes from here to here, so it doesn't leave the spinal cord at all. Okay, that's an interneuron, and it's, remember, interneurons stay inside of the CNS, which of course includes the spinal cord, and it has not left, right? Okay, and then the interneuron will relay its response to a motor neuron, and notice that there's multiple synapses over here, here, and here, all from one motor neuron, and that will cause the muscle to contract. And this is kind of the short version, but the fact is we're looking at three neurons. The sensory neurons that carry information to the central nervous system, interneurons that process information within components of the CNS, and motor neurons that take information away from the central nervous system. Okay, so we discussed neuroglia cells, and we discussed that their job is to support and protect axons, right? And then we also said that they form myelin. So that's an important factor. So let's go ahead and take a look at what we mean. All right, now first of all, this is a full neuron. It's not a neuroglia cell. Now, okay, so why would we put this picture here? Because it's completely relevant. There's two types of neuroglia cells, okay? The first one, these are called Schwann cells. Schwann cells. And the second are called oligodendrites. Schwann cells, now their job is to form something called the myelin sheath, and it very specifically only forms in the peripheral nervous system. Okay, the oligodendrites, they make myelin, okay, in specifically the CNS. So what is myelin? That's the question. All right, now if we look at this, this image, here's the central body. These are dendrites, and then we discussed how neurons, how the axon carries information away. Okay, now some neurons have something called myelin. And now myelin is, if you look closely at this picture, they're layers of this specialized type of membrane. These are called myelin sheaths. Okay, and what they do is they insulate all of this. So all of this is electrical impulses. And when we have myelin sheath surrounding, okay, in a full clump or a full cell of it, so we have the entire myelin structure, okay, all of the layers plus a nuclei. That is a Schwann cell, and its job, the Schwann cells in the PNS, is to of course form all of the myelin sheaths that we see here, okay, but it also saves the neuron energy and it speeds up the impulses. So instead of an impulse moving straight down this long neurons, because some of our neurons are the length of our leg or the length of our arm, and so how do we get these signals to move so fast? This is one of the ways, by insulating these electrical signals, kind of like the way an electrician or just actually, right, any kind of electrical company, when they make cords, you don't see all the electricity, right? It's wrapped in this specialized kind of rubber encasing. Well, the Schwann cells behave that way, except they don't cover it in one strip. They cover it in these kind of units, which are called the Schwann cells. Okay, so what ends up happening is instead of the signal moving down in a straight line, the signal will jump from node to node to node to node, and then this happens. This sends the electrical signal down very, very quickly. Okay, so this is more anatomy that we need to know. When we have a cell that is myelinated like this little package of myelin is called the Schwann cell. Okay, now the space in between that is not myelinated, like right here, that is called the nodes of Ranvier. Okay, very good. All right, myelin sheath give the brain a very distinct appearance. And not just that, it's not only the appearance, it's the fact that it creates something called white matter. So our central nervous system is composed of two types of nervous tissue. We have gray, now we're talking about tissue at this point. We have gray matter, okay, and white matter. Okay, so if you look at the brain here, so we cut the brain and you can clearly see this part is dark, that's gray matter. The inner part, all of this inside, is white matter. The reason why gray matter is gray is because it has neurons that do not have, that are myelin. They're non-myelinated axons, so they're just straight across, there's no myelin. Okay, the white matter contains neurons that are myelinated, so that gives it the white color. All right, so this is two neurons. This is the unmyelinated neuron, okay, no myelin, just a straight axon. And then this is a myelinated neuron. So something that has the myelin sheaths is going to have the nodes of Ranvier with an unmyelinated neuron will not. Okay, so how does a neuron generate signals, or this electrical impulse? All right, so this is actually super interesting. Now neurons make, or they generate and transmit, something called an action potential. It's an electrical impulse, and this is how the nervous system communicates throughout the entire body. Now, And of course, the communication is incredibly fast and effective because it's happening through action potentials, aka electrical impulses. Okay, so let me just kind of re-emphasize this. Okay, the information always flows this way from dendrites to cell body across the axon. Now, if it's an unmyelinated cell, it's just going to, the signal will just pass through the axon. Okay, and those kind of take a little bit longer. Now, if it's myelinated, it's going to jump from one node to the next node of Ranvier, to the next node, to the next node, and the signals that are jumping or being transmitted, these are called action potentials. All right, so we're going to learn about exactly what an action potential is. Now, before we can do that, we have to learn about a couple of things. The first thing that we're going to learn about is called the resting potential. So it's important to understand that when an axon is a very special and unique cell, it's different than any other cell that we have in the body. Now, inside of a neuron, the interior of the cell, okay, it has a voltage. It's actually a millivoltage, but it's a negative. It's a negatively charged interior, and that is normal for a neuron that is not conducting electrical impulses, that is at rest. That's why it's called resting potential, because it's at rest, it's not doing anything. So for a regular cell neuron to just be normal and be in its to just exist and do nothing, okay, it has to have a voltage on the inside that measures at exactly negative 70 millivolts. Okay, now we mean negative 70 in comparison to the exterior of the cell, so versus the outside of the cell. All right, now how is this even possible? Well, our body's genius, number one. Number two, now these neurons have to stay this way when they're at rest, or else, this is something that is so delicate that it could, anything that throws off your nervous system can cause a seizure, it could cause a heart attack. They have to stay this way. They have to. There's no other way. So I want you to know this number right. Oh, I don't know how to erase that. I want you to know this number right now and keep it in mind. When a neuron is at rest, it has to always at rest be at negative 70 millivolts. If we were to take electrodes and measure it, that's what it should be. Okay, and that's normal. And the way that it is maintained, so it's not exactly, it doesn't happen by magic, right? So we have a pump called the sodium potassium pump. Its job is to establish and maintain resting potential. So it maintains resting potential by pumping out sodium ions. So we have ions inside the cell, outside of the cell, but because the sodium potassium pump is designed to pump sodium out, and it pumps potassium into the cell at a disproportionate ratio, it maintains the negative 70. Okay, so this is what I mean by the disproportionate ratio. So this is a sodium potassium pump. Okay, and it's a very generic diagram. Now inside of inside of the axon, so inside of the cell, the axon of the neuron, it's at rest, so it's not doing anything right now. Okay, it has to measure at negative 70 millivolts. So if I were to take a voltage meter, that's what it would measure at. And this is only possible because of this. The sodium potassium pump, what it does is it pumps 3 sodium out. Sodium is positively charged. So if you're pumping something positive out, you're creating a negative environment in here, and that's why you see all these little negative signs, right? Okay, it pumps 3 sodium out for every 2 potassium that's brought in. And then, of course, there's other factors. There's negatively charged proteins and things like that. Okay, but because of this disproportionate pump, this is always when it's at rest, a negative 70. Okay, and this is at rest. Now, things change when this is at rest. Sorry, this is very sloppy. Okay, either way. Now, there are channels, such as this one. This is a sodium ion channel, and they help maintain the resting membrane potential of negative 70 by, if it becomes too positive, for example, potassium ions will move out. They will just diffuse out naturally. And if it becomes too negative, for example, then sodium will diffuse in to maintain this number, negative 70, the resting membrane potential.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
All right, so what happens when it's time to transmit an electrical impulse? Well, it doesn't happen randomly is incredibly organized, and it happens in a sequence of 3 events, and they are as follows. Okay, and I'm going to show them to you in a visual form, but I want you to see these terms. Okay, first, depolarization occurs. Okay, when something is depolarized, it is becoming more positive, and the only way to change the polarity of that -70 is by moving sodium ions into the cell, into the axon, really. Okay, and the reason why that happens is because when the initial signal comes in, it's going to open up a chain of sodium ion channels. And so, therefore, the opening of the sodium ion channels is going to lead to this influx of sodium ions. And sodium ions, don't forget, are positive. So if we have a bunch of sodium, pouring through these channels and they're coming into the axon, they are going to take the voltage of the axon from that rest of -70 to a positive 35 millivolts. Okay, and that is called depolarization. Now, this can't go on forever. It has to stop, and that's when repolarization occurs. Now, repolarization involves the other ion, the potassium. All right, so what's going to happen here is that potassium ions are going to repolarize the cell. Potassium ions will move out of the axon, and because of the fact that potassium channels are going to open, okay, and sodium channels will close. And now potassium is going to move out of the cell, and it will take the positive 35 millivoltage back down to 70. And actually, it's going to bring it much lower than 70, which I will show you. And we rely on this third phase, which is called reestablishment. And the job of the reestablishment process is to return the neuron to rest. And that is done only by the sodium-potassium pump. Okay, so what does this all mean? That sounds like a bunch of numbers and gibberish. Well, it's not. It's actually an incredibly sophisticated process. So earlier, we talked about the inside of the cell has to be at -70 millivolts, correct? Now, this is rest, okay? Now, to elicit a response, okay, there has to be a stimulus. So a stimulus has to come in and stimulate that neuron with a signal, okay? And if the signal is strong enough, it will cause this change. It's going to cause some sodium channels to open up, and sodium ions will move just like in this picture here. Sodium ions are going to move into the cell. Now, it has to reach this measurement though, threshold. Okay, threshold is going to allow a full depolarization. Now, if the stimulus that's coming through is not strong enough to reach threshold, then it cannot the whole action potential cannot occur. All right, so basically, let's say it's strong enough to bring it up to threshold, then we're going to have a full depolarization process. Okay, depolarization is sodium channels opening up, sodium diffusing into the cell, and it's going to, because sodium is positively charged, it's going to take the voltage of the inside of the axon from negative 70 past threshold all the way to positive 55. And that is the beginning of the action potential. Now, this cannot go on forever, and we need the next step to happen, which is repolarization. Repolarization, all of the sodium ion channels need to close. So here, sodium ion channels are open, they're flowing in, now they need to close, okay? And what's going to happen next is that the potassium ion channels are going to open and potassium is going to leave. That means that positive ions are leaving. So if we're looking at the voltage, then on a scale, then we're going to see this whole thing drop from a positive charge to a negative charge. And actually what happens here is it dips down past negative 70, and we need reestablishment, which is step number 3. Reestablishment means reestablishing normal, reestablishing negative 70. That's the normal. And that only happens because of these guys, the resting membrane potential. So reestablishment kicks in, and then we're back to normal. Now, during that time, during those 3 steps, that neuron cannot receive another action potential, and that is a full action. Okay, so we're continuing with action potentials, and now this is an incredibly important figure because what we're looking at here is the measurement of the action potential, the full action potential, and actually other things that occur within a axon. Okay, so let's go ahead and start with the table. Now I want you to look at the x-axis and see that this is, the x-axis is measuring time in milliseconds. So this is a measurement of a neuron, in the axon of a neuron. And on the y-axis, we are measuring the membrane potential in millivolts. Okay, and notice that the numbers are negative, and then here's 0. All right, now, of course, remember, resting membrane potential is at negative 70 millivolts. And we'll go ahead and trace that over. And what we're looking at here is a stimulus coming in. All right, but it's not a very strong stimulus because it causes a slight depolarization, which is actually called a graded potential, a depolarizing graded potential. Okay, now it's not strong enough to create a full action potential, so it goes back down. Now you see a second stimulus right here, and what's happening is it causes a slight hyperpolarization. Okay. And then we see a couple of impulses, 1, 2, and it creates something called summation, which is just, it's a stimulus, and then there's 2 back to back, and basically they're not strong enough to get us to what is important here, threshold. An action potential cannot occur unless the stimulus is strong enough to bring the voltage to at least negative 50. And I want you to remember those numbers. Threshold is at negative 50. Okay, so threshold is the minimum stimulus required to initiate a full action potential. All right, so the stimulus has to be big enough to bring the membrane potential from negative 70 to negative 50. Okay, so here it is. The multiple stimuli have hit, and it brings the the membrane potential up, and then it hits threshold, and that is the point where a full action potential kicks in. Depolarization, then repolarization, then a slight hyperpolarization and reestablishment. Boom. And that is an action potential. Now, this slide it reiterates what I just discussed about graded potentials. Now, graded potentials, they can, we saw a depolarized graded potential, a hyperpolarized graded potential. Now, when these graded potentials reach the trigger point, our threshold is when an action potential occurs, and only when. Okay, now let's actually move to the next slide. Okay, so more about action potentials. Now, here's the thing with action potentials. An action potential is always the same. So, meaning that let's say that you get this incredibly strong stimulus. It's not going to create a higher peak on the action potential graph. It's impossible. What actually happens is that the stronger the stimulus, the more action potentials will occur per unit of time, meaning that they're going to be exactly the same measurement, but they're going to be fast, back to back to back to back to back. All right, so the speed of the action potential, though, is always the same. It can be different in different neurons, however, it's going to follow that exact same pattern.
The nervous system uses electrical signals, called nerve impulses, to move information along the length of the neuron. In the experimental setup shown here, we are using a voltmeter to detect the difference in electrical potential between the inside and outside of the axon of the neuron. When the axon is not conducting an impulse, the voltmeter records a potential difference across the membrane of about -70 millivolts. This reading indicates that the inside of the axon is negative compared to the outside. We call this the resting potential because the axon is not conducting an impulse. Now let's take a closer look at the structure of this membrane. Remember that cell membranes may contain channel proteins that allow the movement of specific molecules into or out of the cell. In the case of the neuron cell membrane, we are going to focus on two channels, the sodium or Na+ channel and potassium or K+ channel. Under resting conditions, the concentration of sodium ions is greater outside the axon than inside, and the concentration of potassium ions is greater inside the axon than outside. This is because sodium-potassium pumps, which are not shown here, actively transport 3 sodium ions out of the cell for every 2 potassium ions that are pumped into the cell. Now let's see what happens when a nerve impulse, also called an action potential, reaches a section of the membrane. Action potentials occur when a stimulus, such as touch or heat, causes the membrane to depolarize. Depolarization is the rapid change in the electrical potential across the membrane. It is important to note that the action potential is an all or none event. If a stimulus causes the membrane to depolarize to a certain level, called the threshold, an action potential occurs. There are no partial action potentials. As an action potential begins, the gates of sodium channels open first, and the sodium ions flow down their concentration gradient into the axon. Since the sodium ions have a positive charge, once they move inside the axon, the membrane potential changes from -70 millivolts to +35 millivolts. At around -55 millivolts, the threshold is overcome, and this becomes an all or nothing event. This sudden rush of positive sodium ions into the axon is called depolarization, because the charge inside the axon changes from negative to positive. Shortly after the sodium channels open, the gates of the potassium channels open, and potassium ions flow down their concentration gradient to the outside of the axon. Because potassium also has a positive charge, as these ions move outside the axon, the action potential changes from +35 millivolts back to -70 millivolts due to the loss of positively charged ions from inside the cell. This is called repolarization because the inside of the axon resumes a negative charge as the potassium ions exit the axon. The action potential travels down an axon one small section of membrane at a time. The depolarization and repolarization of one section of the membrane acts as a stimulus to the neighboring sections of the membrane. So what keeps a signal from moving backward? As soon as an action potential has moved on, the previous section undergoes a refractory period, during which the sodium gates are unable to open. Because of the refractory period, the action potential cannot move backward and instead always moves in the same direction. When the refractory period is over, the sodium-potassium pump works to restore the previous ion distribution by pumping sodium ions outside the axon and potassium ions inside the axon. As we can see here, for each small segment of the axon, the entire process of depolarization and repolarization of the membrane can take just a few milliseconds. Once the refractory period is passed, this section of the membrane is ready to transmit another action potential.
The next part of this chapter, we're going to talk about something called synapses and synaptic transmission. All right, so let's start with definitions. A synapse. Okay, a synapse is very simple. It is the place where two neurons connect, where two neurons meet. It's a specialized junction between the axon of one neuron and the cell body or dendrites of a second neuron. All right, so synaptic transmission is pretty amazing. So now the way that neurons work is there is electrical signaling, right? And that's the action potentials that we just discussed. And then there's synaptic transmission, which is chemical signaling. So our nervous system is incredibly sophisticated, fascinating. Now, transmission of impulse from sending information from one neuron to a second neuron is going to be a synaptic transmission. However, it is very unique because it involves chemicals called neurotransmitters. All right, so this is synaptic transmission, and it looks like a lot, but I'm going to break it down for you guys so you can understand. Now, remember, neurons are connected to one another. They are very much connected, and so basically, what we're looking at here, this is one neuron, okay, and it wraps all the way around. All of this belongs to one neuron. Now down here, this is a second neuron, and it's going to receive information from the first neuron. So let's start with the beginning. This is called a presynaptic neuron. Okay, down here, this is a postsynaptic neuron. Go ahead and label that. Doesn't seem to be labeled, but this is a postsynaptic neuron, the one at the bottom. All right, so now we've discussed action potentials, and they have a huge role here, but this is where the electrical impulse changes from electrical to chemical. Now let's identify some major factors. Okay, first of all, let's see here. Where should we start? All right, now I'll just start from the very beginning. Now, what's coming through the very first step in synaptic transmission is the fact that a neuronal impulse, an electrical impulse, is coming through. And the way that it's going to change into a chemical impulse or a chemical transmission is very strategic. So this is what happens. Now, if you look inside of the neuron, it's not empty. In fact, it has these little vesicles. Okay, can I draw on that? 1 second, let me get my pen back. Here we go. Okay, these vesicles. These are synaptic vesicles, and guess what? They are filled with neurotransmitters. Now, those are chemicals. We have various types of neurotransmitters, but that is something that I will discuss in upcoming slides. Okay, so this is how it all works. The nerve impulse, the electrical impulse comes in, okay? And it's going to cause these channels. This is a channel, this is a protein channel, and it's a very specific type of channel. It's called a voltage gated calcium channel. Calcium has a huge role in the body, including here and in the muscles. All right, now these are voltage gated, meaning that they open up based on a voltage, an electrical voltage, which comes through nerve impulse. Okay, and it's going to cause these channels to open, and calcium is going to move down its concentration gradient through diffusion and diffuse into the actual synaptic end of into the cytoplasm, into the cell itself, into the synaptic bulb. Okay, now this calcium influx has a direct effect on the cell. What it actually does is it basically instructs the synaptic vesicles that are carrying neurotransmitters to fuse with the membrane through exocytosis. Now, I hope you guys remember exocytosis, because this is the time where all of that stuff that we learned is going to start coming into play. Okay, so this vesicle will fuse through exocytosis and release the neurotransmitter into this space. This space is called the synaptic cleft. Okay, this neurotransmitter will bind to receptors on the postsynaptic neuron. Oh, I just realized that it's actually written right here, but that's fine. Okay, now there's receptors, and these are called ligand-gated channels. A ligand is in reference to a molecule such as a neurotransmitter. Okay, so, and what that means is that the ligand or the correct neurotransmitter has to bind, like right here. See how these guys are bound? Okay, and that is going to open up the channel, and once the ligand binds to the gated channel, it's going to open up the channel. And then the next thing is going to cause a response in the next neuron. So that's called the postsynaptic potential, and it's going to cause some type of nerve impulse. Okay, it can actually do two things. It's either going to, depending on the type of neurotransmitter, it can either cause the postsynaptic neuron to depolarize, which means that it's going to cause a full-on action potential, right? Or it can cause hyperpolarization. Okay, or it can cause hyperpolarization, which means that it in fact would not, if it hyperpolarized, that means that the neuron would kind of just turn off. It wouldn't send off a signal. All right, so that's going to be based on what type of neurotransmitter is involved here. And that's where this comes in. So we have two major categories of those neurotransmitters. We have excitatory neurotransmitters. Okay, excitatory neurotransmitters will cause the postsynaptic cell to depolarize. Okay, inhibitory neurotransmitters will cause the postsynaptic cell to hyperpolarize. All right, so I think that neurotransmitters are some of the most fascinating aspects of the nervous system, and pretty much actually the entire human body because they control everything. So, when they say that we're made of chemicals, yeah, it's very true. And on top of that, now we have this entire set of neurotransmitters that control a lot of things. So let's look at some of them. Acetylcholine is a neurotransmitter, and it's an excitatory neurotransmitter in some cases. For example, it's involved in skeletal muscle contraction. That's excitatory. However, there's certain times that it can be an inhibitory neurotransmitter depending on the receptors. Okay, so it acts at the neuromuscular junction where neurons meet the muscle. It acts inside of the brain as well. Okay, oops. So the next one is pretty interesting. It's norepinephrine. Norepinephrine is excitatory for the most part. It plays a role in your emotions. Okay, and typically this is something that actually is, a good thing. It makes you feel good. Right. And the next one is very interesting as well. It's serotonin. Serotonin is going to be released in the brain, in the spinal cord. It usually has an inhibitory effect. It has an effect on a person's mood. It has an effect on your sleep cycle and appetite. So people with low serotonin, people with low serotonin are basically people with low serotonin have depression. Okay, and this is a very common thing in especially in our society. And sometimes they're just, they have low serotonin levels genetically. Sometimes it's something else. And then, of course, we have medications for that. But that's a kind of another aspect. So let's keep going. Okay, dopamine. Okay, dopamine is pretty interesting as well. This is they call it the love neurotransmitter because this is the one that makes you feel really good, and your brain releases dopamine when you, okay, let's say you drive by, you have a favorite restaurant and you see an advertisement for that restaurant, or you drive by and you see a sign or an advertisement for it, right? And you feel like you start to salivate. It's because your brain is releasing this dopamine, and dopamine has a huge role in emotions, and so that's why they call dopamine the love neurotransmitter. Okay, and there's also, actually a lot of people agree, and it's been proven that dopamine is released when people fall in love. All right. Glutamate. Glutamate is pretty interesting. Glutamate is an excitatory neurotransmitter. And if our nervous system was a car, then glutamate would be the gas pedal. So it turns things on. Okay. And using that same analogy, if our nervous system was a car and glutamate is the gas pedal and turns things on, then GABA, this is GABA right here, okay? GABA would turn things off. Okay, what just happened? All right, hang on one sec. All right, I'll just move this over here. I'm just going to move it over here. So GABA would turn things off. GABA is inhibitory, and it's absolutely necessary for life. You have to have gabapentin. I'm sorry, not gabapentin. That's a medication. You have to have enough GABA and enough glutamate for a perfect balance. Just like a car, you need to have that gas pedal, brake pedal, gas pedal, brake pedal. Okay, so we need glutamate to turn things on, and we need GABA to turn things off, inhibitory. Okay, without a perfect balance of these two guys, a person can have a seizure. Okay. Endorphins. Okay, endorphins. Endorphins are natural. Endorphins are released when you exercise and then you start feeling really good at some point. It takes a long time and, you know, quite a bit of dedication to get to that point where you really feel good exercising. Your body starts releasing natural endorphins. But what they actually are is we have a natural opiate system, and our endorphins bind opiate receptors and they inhibit and block pain. So we have them in the brain, in the spinal cord. Okay, those are the major ones that I want to discuss. All right, so this is an interesting aspect. So cocaine addiction. All right, or cocaine in general. Okay, so this is how some drugs work, and actually quite a few drugs work by acting on synaptic transmission. Okay, so here we go. Now, what's going on here is, okay, there's the electrical neuronal impulse, right? And this neuron is releasing the chemical dopamine. Dopamine is the love, happy, euphoric feeling that is actually created from too much dopamine, right? Okay, and when a person does, if a person were to do cocaine, the way that cocaine behaves is as follows. All right, so typically, you know, neurons are released and then they're used to a certain extent and then they are taken back up. This is called the reuptake. So we can't just have neurons floating around forever. That's not good. Okay, so this reuptake process happens. Well, guess what? Cocaine blocks the reuptake of dopamine and in fact signals or triggers the release of dopamine. So there's this big influx of dopamine, and that is what causes a person doing the cocaine to feel the euphoric effects of the cocaine. It's really the dopamine. And yes, there's other things involved like norepinephrine, but it's mostly dopamine. Okay, now this is an important factor here. Oh, really quickly, let me just show you guys something. All right, so there's always a reuptake process. For some reason, this picture is lacking the reuptake channel. Okay, so I talked about serotonin earlier, serotonin and depression. So we can actually use the way that neurons behave to our advantage. So when we have a person that has depression, the doctors will prescribe something called an SSRI. An SSRI is a selective SSRI. I think I put 3 S's. SSRI. Okay, it's a selective serotonin reuptake inhibitor because normally the chemicals are the neurotransmitter chemicals are released, and then when there's too much or they're taken back up. When they're used, they're taken back up in this reuptake process. This is a normal thing. They do this, our neurons do this all the time. This is called reuptake. So somebody that has low serotonin will be prescribed an SSRI because SSRIs block the reuptake of serotonin. And then ultimately the SSRI will increase, because they block the reuptake of serotonin, we're gonna increase the levels of that person's serotonin. And having high serotonin or higher serotonin, it is going to have a positive effect on their mood. Okay, so that's how we actually use the nervous system quite a bit to for medications. For example, GABA. GABA is designed to, we know GABA is an inhibitory neurotransmitter, and it calms the nervous system down. So if somebody has high anxiety or a highly active nervous system, something that's in this that behaves like GABA is is going to be something like Xanax or Valium, one of those medications. Okay, so that's one aspect. There's a lot of things here that we can manipulate. We know that we have opiates that are used for pain in the hospital and after surgery, and they behave like the endorphins, and they bind to opiate receptors and inhibit pain. So, all right, so ultimately, our knowledge of neurotransmitters is incredibly important because it leads to our knowledge of how medications work. And if you're going into the medical field, this is something that you're going to learn and relearn and learn again. All right, so let's go on. Now, let's look more into neurons. So we have postsynaptic neurons that can integrate and process information. We know that, but what's interesting here is that we can have one neuron act on multiple neurons, and multiple neurons can act on one neuron. Okay, that's an important aspect there. All right, so earlier we discussed how the nervous system has two primary parts: the central nervous system, the peripheral nervous system. Remember, the CNS is the brain and spinal cord, receives, processes, and transfers information. The peripheral nervous system is all of the nerves outside of the central nervous system, and it has two divisions, sensory and motor. So we've seen this slide before, so I'm going to move quickly on. So I want to show one diagram here that's pretty straightforward, but basically, here's the central nervous system, right? It's just kind of brain and spinal cord, so it's this little box. And information, all of this down here is going to be the peripheral nervous system. So sensory input. Okay, these are signals from the environment, like smell and touch and vision, right? Okay, signals from your skin, tendons, muscles, from internal organs. Okay, all of that is sensory input, and it goes to the spinal cord, and it could get sent to the brain. It depends on the signal, and the brain will send a response back, and that response is carried out by the motor output. Okay, so then the information comes in from the sensory, okay, and the brain and spinal cord will make a decision as to what needs to happen in response to that signal, and then that's the motor response. So it can do quite a bit actually. It can control muscle contraction. It can control the autonomic division. Okay, the autonomic division of our body is all of the stuff that we cannot control. So think of autonomic like automatic, but it's not automatic, it's autonomic. So autonomic control is smooth muscle contraction. You don't control your stomach and your GI system contracting. You don't control your heart contracting. You don't control your glands. You have no control over that, and that is the autonomic system. And autonomic is divided into parasympathetic and sympathetic nervous system. So the nervous system is quite complex, but if you break it down, it can be manageable. All right, so let's look at the somatic versus autonomic. Okay, I think we actually, okay, well, fine. All right, so let's look at this a little closer. Somatic. Somatic is pretty straightforward here. It's going to be just a direct action on something like a muscle, like that we have control over. Okay. And acetylcholine is directly involved in muscle contraction. The autonomic system is something that we don't have control over. And it's going to be, things like this is a heart muscle, so like the heart contracting, smooth muscle contracting, our glands, our endocrine system, for example, releasing hormones and things like that. We don't have control over that. All right. The somatic division of the PNS, again, controls the skeletal muscle system, and for the most part is voluntary. And then, yeah, we do actually have reflexes. And ultimately, a reflex is involuntary. Now, the autonomic system, again, controls the autonomic functions, automatic functions, and it's divided into the sympathetic and parasympathetic division. Okay, so for every action, there's an equal and opposite reaction, right? So, in other words, the sympathetic division is more of it turns things on, I guess you could say, and then the parasympathetic is more of it relaxes things or turns things off. And it's a lot more complex than what I'm discussing here, but for this course, we're not going to go much further than this. So you can look at it this way. The sympathetic division is directly involved in your fight or flight responses. So the primary neurotransmitter is going to be norepinephrine, one of the primary transmitters, and it prepares for emergencies. The parasympathetic is going to be the primary neurotransmitter is acetylcholine, and it's involved in more relaxing and turning things off that the sympathetic division has turned on, and they completely oppose each other. Everything that the sympathetic division can turn on, the parasympathetic can actually combat that or do the complete opposite, and they work together to maintain homeostasis. Okay, the next part of this chapter is focused specifically on the central nervous system, which of course is the brain itself and the spinal cord. Now, the central nervous system is where our sensory information is going to come into play, where signals from the PNS comes here, and it's received here. And in turn, of course, there's the central nervous system is sending out motor signals, or it has motor function and control over the body. Now let's talk about how the central nervous system is protected. Of course, we have our scalp, and then right underneath we have our skull. Okay, and the vertebral column is not depicted here, but that's very clear that the vertebral column protects the spinal cord. Now let's look at what's underneath the skull itself. Underneath the skull, we have the meninges. Okay, so the meninges is composed of several layers. So we have the dura mater. Okay. We have the pia mater, which actually encases the brain itself. And then we have the arachnoid, and it's called the arachnoid because if it's looked at under the microscope, it looks like a spider, or well, spidery, I guess you could say. Now we also have something called the blood-brain barrier, which is not depicted in this picture, but the blood-brain barrier protects the brain because, of course, just like every other organ in the body, we need the brain to have a constant supply of oxygen, nutrients, and CO2 has to be taken away. Our brain burns something like around 128 grams of glucose a day in cellular respiration. Now, the blood-brain barrier ensures that if pathogens and certain chemicals are indeed in the blood, which they tend to be, they cannot cross the blood-brain barrier. Now, yes, there are, a few that can can breach that barrier and cause an infection in the brain, which is rare and very scary, but we do have this defense, the blood-brain barrier. Now, what's important to see here is see this blue part, okay? That is called the cerebral spinal fluid. And then this is the brain tissue itself. All right, so let's go on. Now, the central nervous system is the brain and spinal cord, and I want to point out a couple of things. First of all, the brain contains these hollow cavities called ventricles. Now, they're not empty because what they indeed are carrying is that cerebral spinal fluid. Okay, and what's interesting is this. So this is, we're obviously inside of the brain. Okay, we're inside of ventricles of the brain. And if you follow these arrows, okay, notice that the cerebral spinal fluid connects all the way around into and it connects to the spinal cord. So this is a direct link between the brain and the spinal cord through the cerebral spinal fluid, also called the CSF. So basically these ventricles are this continuous canal or pathway with the brain and spinal cord. And ultimately what it does is it produces and stores the ventricles produce and store the cerebral spinal fluid, which is responsible for bathing the CNS. And it's a shock absorber. All right, now let's check out the brain. Now, the brain is probably the most complex organ of the entire body, but it is manageable, and it is possible to break things down and learn them on this level, on a biology 100 based level. All right, so let's start with this. Now, the brain is the boss. It's the command center of the entire body. Okay, so that's the beginning. We can break the brain down into 4 major divisions. The first is called the brain stem. Then we have this section of the brain called the cerebellum, the diencephalon, and the cerebrum. And each one of these has incredibly vital roles, and this is minimal information, but it's significant information. And I've definitely minimized these functions. Okay, so we'll start with the brain stem. Its primary job is to regulate vital functions, such as autonomic functions, and we'll talk more about what that means. And it's a relay station. Yes, it is. It relays information to higher regions of the brain. The cerebellum, it controls your basic posture and balance and skilled movement. The diencephalon is broken into several divisions, and so is the brainstem actually, but one of its main jobs is to maintain your homeostasis, which is essential for a healthy life. It also integrates information that's coming in, and it also it integrates that information, and then it also can pass on information to higher brain regions. And then it's responsible for your sleep and wake cycle. Okay, the cerebrum is the largest part of the brain, and we're going to look into that as we go through. But here, just for simplicity, in the cerebrum is going to receive information, right? And it's going to integrate sensory input, and it's going to determine complex behavior. All right, so let's go ahead and start with the brainstem. So this is a great picture of the brainstem because is highlighted here in this kind of yellow reddish color. And now if we look closely at it, the brainstem is made of several parts, 3 major parts. Okay, let's start from down here. Here is the spinal cord. Now the spinal cord has to end, and it does. It ends in the medulla. So the medulla is this region of the brain. And then see this part that it's got these kind of stripes. Okay, now if you were looking at a real human brain, you would actually see these stripes. And I'm going to show you, it will show you some pictures. Okay, so that's how you identify the pons, just a little hint. Okay, and that is called the pons. And right on top is this little tiny knot. And notice that it also has striations, but they are vertical. And that is called the midbrain. All right. Okay, the next section we're going to talk about is the cerebellum. So the cerebellum is right here, and it's kind of, it almost looks like a little brain, and it actually is called that. It's called the little brain or the small brain because it's kind of this own unit, but of course it's directly connected. Now the cerebellum receives 3 types of information. Critical information for your equilibrium. Okay, and basically information on current movement. So if you move your head from left to right, your neck, your limbs, your trunk. Now, how do you stay balanced? That is the cerebellum's job. And then it will also receive information. So it's receiving equilibrium information, information on your movements within the body, and then information from the cerebral cortex, which we will get to. Now, its primary job is to make sure the body is balanced and organized in movement. Pretty simple. If we look at the cerebellum, this is a real brain actually just a picture of it zoomed in. Okay, so let's check this out. So this is, if we cut a brain in half, and this is the cerebellum. This is what it looks like. It has these little, folds, but if we were to cut it, you'd see some interesting structures here. So see how there's these kind of branch-like structures, okay, extending out. Okay, those are called the arbor vitae. Okay, now, and they're white. Now the darker part, the gray matter basically, is called the cerebral cortex. All right, and what's important as well, of course, there's an anterior posterior lobe. What's important to note also here is that, remember, we discussed briefly ventricles. Now there's several ventricles in the brain, and one is right here in between the cerebellum and the brainstem. It's called the 4th ventricle. Okay. So I like to show several pictures of the brain, just to give you different perspectives, because ultimately the brain is going to be basically identical, and if you look at it through the lenses that are required to just see these important structures. Now, even though this is a real brain and this is in animation, we can still see the same shape of the cerebellum. Okay, this kind of little heart shape, I guess you could call it, right? Okay, and then we can see the arbor vitae, we can see the cerebral cortex, and then of course there's that ventricle. So I want you to know how to identify the regions of the brain with various images that we are using. All right, let's talk about the diencephalon. The diencephalon, it's basically encased in the cerebral hemisphere of the brain, which is the biggest part of the brain. and it's primarily three structures. Well, it is three structures, not primarily. The thalamus, and you guys may have heard these terms before, and we're going to talk about what they do. They have a big job. Thalamus, the hypothalamus, and then we have the pineal gland. And it's important to know exactly where it is. I'm going to show you where, but the third ventricle, the diencephalon borders the third ventricle, and it's made mostly of gray matter. Okay, so this is the diencephalon. So let's look at each structure independently. All right, so if you see, I like to teach the hypothalamus like this. Okay, so here's the hypothalamus, right? And if you look at a brain from this angle, meaning what we're looking at right now is a mid-sagittal cut of the brain, and we're looking at the right side of the brain. And if we look at that, this is the hypothalamus. It's this structure, and you can, a good way to remember it is it kind of makes an H, right? And it's directly connected to this gland that we will discuss later, called the pituitary gland. Okay, now this roundish kind of area, okay, that is the thalamus. Okay, and then right behind the thalamus, or near the thalamus, is the pineal gland. Okay. So that little tiny gland, it almost looks like a little piece of rice. Do not mix up the pineal gland with the pituitary gland, which is big, and it has two lobes, in fact. It's a big one. All right, the diencephalon, the thalamus. Okay, the thalamus itself, okay, remember this is the thalamus here. Okay, the thalamus makes up 80% of the diencephalon. It's relatively big. And basically it's carrying dozens of major nuclei. And so it actually acts as a relay station of incoming sensory information. So remember, sensory information is coming from the PNS, and it's going to go through the brainstem, and it has to eventually go to the thalamus. And the thalamus plays a major role because it receives sensory information, and it's going to relay that information to the correct part of the brain. So every part of the brain communicating with the cerebral cortex relays signals through the thalamus. So the cerebral cortex is the higher region of the brain, but nothing gets through to the cerebral cortex without first going through the thalamus. Okay, now it's capable of doing this because, of course, it has because of axons being long enough to reach the different regions of the cerebral cortex. So that's what it is. It's a relay station. Okay, a lot of people like to say this is the gateway to the cerebral cortex because it basically is. All right, the hypothalamus, which is the one I said looks like an H, right? And it's right next to the pituitary gland, which is the hypothalamus, pituitary gland have a huge role in our endocrine system, in our hormones. And the hypothalamus, not surprisingly, because of the fact of its connection to the endocrine system, controls a lot of the autonomic nervous system, your emotional responses, body temperature, which is basically, let me just put it this way. The hypothalamus helps maintain homeostasis, so your body temperature, hunger and thirst sensations are regulated by the hypothalamus. Certain behavior, your sleep-wake cycles, and then the control over the endocrine system and then it has a role in memory formation as well. So, okay, now this is actually from one of my anatomy classes, this figure, and it really shows in detail how complex the hypothalamus is, with all of the different nuclei and its direct connection to the pituitary gland. And I just want to emphasize that the complexity here is it cannot be understated. But we're going to keep things simple. Now I want to point out another structure. See this little structure? Okay, this little guy right here. Okay, at the kind of actually, if we were to look at this picture, it would be right here. Okay, this is called the optic chiasma. Okay, very good. All right, the pineal gland. Your pineal gland is incredibly important because it secretes the hormone melatonin, and melatonin is involved in sleep. And sometimes if a person is not sleeping well, they could take melatonin supplements, but the secretion of this particular hormone is under the direct control of the hypothalamus, but of course the pineal gland is responsible for that, and it aids in the control of something called your circadian rhythm. So your circadian rhythm is something that has developed since basically, birth, and your body has been able to process light, which equals, daytime, which equals being awake. And it's also this is the whole circadian, a simplified version of the circadian rhythm. And it also has built your brain to understand that dark equals nighttime, which equals sleep. That is, in a nutshell, the circadian rhythm. So, in fact, people that are blind, for example, they don't have, or especially if they were born blind, they have a problem sleeping because their circadian rhythm was never fully built correctly because they can't see light or dark, right? Well, actually, they only see dark. All right. Now, the cerebrum. Okay, the cerebrum is the largest part of the brain, and it has a right and left hemisphere. Okay, this is going to be the right hemisphere over here. This is the left hemisphere. Now, the cerebrum, the right and left side, is connected by these nerve tracts that allow the right and left side of the brain to exchange information right there. Okay, and that's called the corpus callosum. All right, so ultimately, the couple of notes here. The brain, in the cerebrum, the outer portion, see all of that? Okay, I know it's in blue and pink and all that, but ultimately what that is, it's technically, that's gray matter. Okay, and then that's called the cerebral cortex, and the cerebral white matter, well, the whole thing is the cerebrum. The cerebral white matter is inside. Okay. So I want to point out the corpus callosum in this perspective, because if we cut the brain and we look at, we do a mid-sagittal section of the brain, this is the corpus callosum. So it's not just, a couple of nerves, it's this full structure. Okay, see, and it kind of looks like a little, to me, I always imagine it looks like a little, like a finger, like a hooked finger or something like that, right? And it allows, and this is the whole thing. Okay, that's called the corpus callosum, and it has a huge role in making sure that the right and left side of the brain have communication and can signal back and forth. All right, so while I have this image here, I want to point out some very important factors. Okay, now so far we have discussed the medulla. Okay, on a real brain, this is what it looks like. Not much, but we know that it's right there. The pons is easy to identify because it's kind of round. Okay, there's the pons, and technically that's the midbrain. All right, let's look at the cerebellum. The cerebellum is easy. There's the cerebellum. Okay, got that. And the fourth ventricle. All right, what about the other ventricles? All right, so let's look at the lateral ventricle. Okay, this is the lateral ventricle. So right under the corpus callosum, see this space? That is called the lateral ventricle. And yes, you do have to know the ventricles, otherwise I would not be covering them. All right, now where are we here? Okay, now let's find the thalamus so we can navigate through the brain. There's the thalamus, right? Okay, and I want you to notice another little tiny ventricle right there. See that little space? That is called the third ventricle. Okay, now there's the hypothalamus. Okay, and looks like I always think of an H. It also kind of looks like a tooth, right? And we actually can't really see the pineal gland on this one, but what you can see is the optic chiasm. And it looks like this little piece. And remember, the optic chiasm is right next to the hypothalamus, because on this side, there's another structure that looks like a little piece of rice as well, and that is the pineal gland. So remember, the pineal gland is next to the thalamus. And the thalamus is, of course, is this area, this roundish kind of area. Okay? All right. Right. Okay, that's good for this image. All right, let's talk a little closer about the cerebral cortex itself. Now we have it's broken into lobes. All right, so we have our frontal lobe. The frontal lobe is incredibly significant. They all are. Now don't forget we have a right and left side. Okay, so there's a this is a right frontal lobe. So if this brain was our brain, then of course this would be the left. Oh, that's supposed to be an L. This would be the left side of the brain, and that would be the right. Okay, so we have our frontal lobe, and ultimately it has a huge role in your motor activity. It's responsible for speech and conscious complex thought. Okay, now right behind it, okay, I'm going to draw this line to cut this up. All right, in blue here. This is called the parietal lobe, and of course we have a right and left parietal lobe, and this parietal lobe interprets sensory information. And a lot of the sensory information coming in comes from your skin. All right, so in the back, this back lobe is called the occipital lobe, and it actually processes vision. So I don't know if you've ever heard this story of this individual who was playing baseball, and a professional baseball player, threw this baseball, and professional baseball players throw baseballs at, extraordinary speeds. And they were hit in the back of the head with the baseball at this extraordinary speed, and they went blind. Why? Because the occipital lobe was damaged. And the occipital lobe is responsible for vision. Now, on the side, we have a right and left side, and this is called the temporal lobe. The temporal lobe, this should not surprise anybody. The temporal lobe is near your ear, right? Your ears. Okay, so it's responsible for interpreting auditory information. It also is responsible for comprehending language and making perceptual judgment. So these are complex thoughts. And that is the cerebral cortex. Now, this is, kind of, some of it is repeat information, some of it is a little more extensive information. This is a great image because we can see, there's the cerebellum, and the cerebellum is the big, massive part of the brain. And, we discussed the different parts, but it's kind of wrapped it up into like language, decision making, conscious thought, vision from the occipital lobe, of course, the brain stem. The midbrain is involved in relays visual and auditory information. Okay, so that's important, and it has a role in coordinated movement as well. Okay, the pons connects the cerebellum, spinal cord with higher centers of the brain. So the cerebellum is coming this way, right? The spinal cord's coming this way, and the pons is going to connect them to higher regions of the brain, and that's an important factor. So here comes the cerebellum, boom, spinal cord. Well, really the spinal cord's way down here. The pons helps to initiate sending these signals up to the correct regions of the brain. Okay, so it'll go through the thalamus too. All right. Either way, this is what we mean by higher regions of the brain, the cerebrum. Okay, it also helps the medulla in respiration. So in fact, your medulla is involved in a lot of important functions, such as breathing, which you may remember from the respiratory chapter. And it also, just to kind of, wrap it up, basically it has a lot of control on the autonomic functions of our internal organs. So our medulla has a huge job. Okay, remember the cerebellum is balance and coordinated, skilled movement, balance, equilibrium. Okay, hypothalamus in a nutshell regulates homeostasis. The pineal gland is involved in the sleep-wake cycle, and then the thalamus is that relay station. It receives, processes, and transfers information. This is a great image to study and to add to as well. All right, the next thing is the spinal cord. The spinal cord is this, it relays information, and it acts as a superhighway for basically for information coming through and leaving between the brain and the rest of the body. So that's, we can kind of leave it at that, okay, and just to kind of keep things nice and smooth. Now, the spinal cord, what's interesting about the spinal cord is the white matter is on the outer portion. So all of this outer portion, all of this, that is all the white matter, right? And then see this kind of butterfly shape. Okay, that is the gray matter, which is the complete opposite of the brain. So I'll quickly remind you that the brain itself, the outer region is gray, the inner is white. All right, so that's just, just a quick point there. Now, of course, the white region has myelinated sensory and descending motor nerve tracts. So it's white because of myelin. Okay, so the gray region is going to carry these cell bodies, dendrites that are non-myelinated, and we'll just leave it at that. What about memory? Okay, so we have both short-term and long-term memory. And basically short-term memory is from working memory. It's information from in a span of just a few hours, and it's stored inside of something called the limbic system, which we haven't really discussed, but it is a part of the diencephalon, the limbic system. Okay, long-term memory is memory from previous days or years, and it actually involves permanent changes to neurons and neuronal synapses within the cerebral cortex. And that information is stored in an area called the hippocampus. The hippocampus is part of the limbic system. All right. Let's talk about disorders of the nervous system. Okay, the very obvious ones is trauma. Physical trauma to the brain or spinal cord can destroy a person's life. I mean, you can become paralyzed if you damage regions. You can, people don't come back from this damage. And then there could be things that are very mild, like a concussion is relatively mild depending on how severe, which just disrupts electrical activity. Spinal injuries, is just impairment of sensation, and it will impair function below the site of injury. All right, now infections. Okay, encephalitis. Okay, encephalitis is inflammation of the actual brain itself caused by a virus that has breached the blood-brain barrier. So remember, it's not impossible for organ invaders to breach the blood-brain barrier, but it's pretty rare. So encephalitis is one. And here, if you look at this image, you can see this inflammation on this side and encephalitis. Okay, now meningitis is inflammation of the meninges. Remember the meninges that we discussed earlier, which is encasing. It's this big, thick, membrane that encases your entire brain. Inflammation of the meninges is incredibly dangerous. You have to absolutely seek medical treatment for any of this stuff. All right, I don't know if you guys ever heard of a couple who had a son, and that son was diagnosed with meningitis, but they were holistic medicine people, and basically they were trying to treat meningitis with herbs and things like that, and then in the end he died, the boy, because of their choices, and they did not seek Western medicine or help until the boy was unconscious. Then they called the ambulance to come, and it was too late. And they were charged with, well, actually, I think they got out of it, but there was a trial for their murder, for basically negligence and the child's death, but I think they got off for it because they had a good attorney. Either way, something like that, there should be a great balance between Eastern and Western medicine, but when it comes to these, inflammation of the meninges or something major, you never go the holistic route. You go to the hospital. All right, rabies. Rabies is an infectious disease that is transmitted through animals. And, it used to be an issue. It's not so much an issue anymore because of the laws that have been set down that basically, people have to give their dogs rabies shots. But it used to be a big problem, and it's an infectious viral disease. and it spreads from the bite of an infected dog to the brain, and it damages the sensory neurons. Okay, epilepsy. When someone is epileptic, they have seizures, and okay, we can, epilepsy is when neurons are firing on and they won't turn off correctly. And things that will fire on a neuron, remember, is glutamate. So not all epilepsy is caused by this, but sometimes people make too much glutamate. So their brain and their neuronal signals are being turned on, and they lack GABA. And so we have to give them benzodiazepines, which mimic GABA and bind to the GABA receptors, and we can help minimize the seizures. Okay, Alzheimer's is a pretty sad, scary disease. Now, Alzheimer's, what actually is happening here is the body, the brain, it stops making acetylcholine, or the production is slowed down, and ultimately it's going to lead to memory lapse and dementia. Parkinson's is the loss of dopamine releasing neurons, and unfortunately, this affects motor function, and people shake when they have Parkinson's. Now, this is extra information, but it's interesting. Now, what does drug abuse do to the nervous system? We talked a little bit about the stimulant and the drug cocaine, and so I'm going to talk a little bit more about drug abuse. Now, of course, drug abuse will alter the mood and emotional state of a person. Now, usually the first time a person tries a drug, they will experience a feeling of euphoria or pleasure. Now, sometimes it really just depends on the person's anatomy. It's also there's definitely that is it nature versus nurture, but some brains tend to be more addictive brains, and they can try this drug and they want to just try it again and again and again and again, and then leads to addiction. Others can try a drug maybe and basically experience it and then walk away without wanting to do it again and again and again. And this is a very huge topic, so I'm just going to leave it at that. But it really does come down to the brain structure and anatomy, and usually what type of household they were brought up into as well. So it's both factors. Now, if a person continues to use drugs, of course they're going to build a tolerance, and they're going to have to ingest more and more and more of the drugs, and they will acquire a physical, psychological dependence, and then of course quitting will be difficult for them. Now, what drugs are we discussing here? All right, so here's the thing. There's two major types of categories. There's stimulants and depressants. So stimulants are going to excite neurons. Depressants will decrease neuronal activity. Now, dopamine is one of the primary neurotransmitters involved in the physical dependence or addiction. So, for example, cocaine increases dopamine. It blocks dopamine reuptake. Okay, and actually, a lot of the drugs they act on the synaptic transmission. So, one thing they can do is cause a neurotransmitter from the synaptic vesicle to indeed move and bind to they will cause the release of the neurotransmitter. So that's what some drugs do. Okay, so especially if it'll act on dopamine, it'll cause this big influx of dopamine, which leads to euphoria, preventing the reuptake. That's a big one. So let's say there's always actually a reuptake channel here. Okay, a lot of drugs will block the reuptake of dopamine, serotonin, norepinephrine. Now, promoting the release of a neurotransmitter, preventing the reuptake of a neurotransmitter. Okay, so we have enzymes that break down neurotransmitters because we're not really supposed to have this huge amount of dopamine. This is not normal. This is like this euphoric state that is basically it's an altered state caused by an external chemical. Okay, so some of the drugs can block the breakdown of the neurotransmitter, or they can behave like the neurotransmitter itself, mimic the neurotransmitter. Okay, nicotine. We'll talk about nicotine quickly. I don't want to spend too much time on it. Basically, everybody knows what nicotine is. Ultimately, it's going to cause the release of epinephrine, okay, in the peripheral nervous system, and it's going to cause dopamine to be released in the CNS. And basically, this dependence is caused from the dopamine, and somewhat from the epinephrine as well. But the dopamine really is it produces that physiological, psychological dependence. And it doesn't release tons and tons of dopamine like cocaine, but it still releases dopamine. Alcohol is an interesting one. Alcohol influences GABA. GABA, remember, is an inhibitory neurotransmitter. So if you're consuming alcohol, after a certain while, depending on exactly what's happening. Okay, really quick. Also, serotonin and dopamine are released, but the main one is GABA. Now, college students and people in general, and alcohol is very acceptable in our culture, and people drink, and it's not a crime. But this is what alcohol does. So it binds to GABA receptors, and GABA is an inhibitory neurotransmitter. So what's happening is these GABA receptors alcohol is binding GABA receptors, and GABA is turning off different neurons, and then it's going to impair motor function. This is why people will fall and stumble and lose their balance and lose their ability to speak when they're drunk, because GABA is shutting down all these neurons, right? Okay, now with the addiction process of it, this is still something that's being researched extensively, but obviously, we know some things. We still don't have a full solution, but there are some treatments for alcohol addiction. Now, alcohol is metabolized in the liver. Everything technically is detoxed in the liver, but alcohol prevents the liver from breaking down fats. Okay, the fat accumulates in the liver after only one night of drinking. This is the first stage of damage, but it's reversible. However, continued consumption causes scar tissue in the liver, which is still reversible. However, long-term consumption at this, depending on the rate, can cause cirrhosis, and that is irreversible. Liver cells die, and that is the end of that. A lot of people, might be candidates for liver transplants, but it is a scary thing. Okay, marijuana, I'll just quickly talk about this. It releases GABA, it releases dopamine. And here's the thing, yes, there's medicinal properties for marijuana, but from the neuronal perspective, from the neuropharmaceutical perspective, which is my perspective, which is my background, what we what has been shown by researchers is that habitual use of marijuana indeed increases dopamine inside of the hippocampus, which is actually what schizophrenia is. Okay, schizophrenia. People with schizophrenia, their brain makes too much dopamine, and specifically the dopamine is concentrated in the hippocampus region. So, that's just an important, factor there. Now, it doesn't mean that for sure, the person's going to become schizophrenic, but what's happening, and this is a fact, is that heavy usage, the brain starts to mimic a schizophrenic brain. Okay, so I'm going to just go over this quickly. We've already talked about cocaine. Crack and methamphetamine behave the same way. They block the reuptake of dopamine. Okay, and so basically, it's going to. Now you don't have here's the thing, you don't have this endless supply of dopamine. So pulling dopamine and dopamine and dopamine and dopamine, of course, there's going to be a crash, because for every action, there's an equal and opposite reaction. You cannot feel so good without feeling just as equally bad in response, okay? So that's that. And ultimately, the thing is it's going to reduce the natural production of dopamine. It's going to create this, tolerance where basically there's going to be an intense craving for the drug. Overdosing obviously can cause seizures, heart attack, respiratory, but long-term use can cause actual brain damage. All right. So this is the brain's activity. Before cocaine usage, it's very active. This is a brain after cocaine activity, and you can see the activity is very, very minimal. All right. Heroin is an opiate. It binds to our natural endorphin receptors. Now, we use opiates in medicine like codeine, morphine, oxycodone, but they're all derived from the opium plant. So repeated usages of an opioid can lead to a pretty terrible addiction, and people have to be careful. Heroin addiction is in every single class in America. There is no area or social class or socioeconomic class that is immune to heroin addiction. In fact, it's seen mostly in the upper class and with celebrities and whatnot. Okay, now overdosing can lead to respiratory failure, convulsions, and death, and this goes for any opiate, even the Vicodin and that sort of thing. Okay, I'm not going to really talk about this one. If you want to read it, that's fine. I do want to talk about the irreversible depression that is caused from long-term usages of any CNS stimulant. In fact, like methamphetamine, cocaine, all that. Releasing dopamine is not a good thing. It's going to cause irreversible damage to the brain, and the brain is not going to be able to function properly and make dopamine properly, and in the end a person is going to be in big trouble, because this depression is not fixable. Yes, we can give antidepressants, but they'll never be the same again.